Okay, good morning, everyone. And we'll continue our discussion today about the immune system. As I just want to tell you that we will just have a discussion when I see you in the new quarter. After the spring break, we will have a discussion about your tests, about your exams. I'm not going to discuss it today. All right, so this is where I left you last time in our abbreviated class because of the projector failing. We were looking at this, the structure and discussing a little bit about the function of the different leukocytes. The leukocytes are the white blood cells, and they are the active living part of the immune system. So it behooves us to spend some time actually seeing what they are. Go back, please. Remember what I told you. We discussed mast cells, which are involved with inflammatory responses. We discussed the neutrophils, which are the workhorses, the soldiers of the immune system. They are the cells which actively hunt down and kill pathogens and also consume debris from damaged cells, etc., etc. We'll have a little bit more to say about them and about their function in a second. So just let me do this. Okay, so the neutrophils are easy to recognize because they have these strange nuclei, these strange lobular nuclei like this. And they are relatively small cells, but they are the most numerous of all of the white blood cells in the blood. The other one we talked about were the monocytes with this large nucleus. They're quite large cells compared to the other leukocytes. Have this large nucleus with a crease in the middle there. And the other group that we talked about were the lymphocytes that we're going to hear a lot about later on, especially when we talk about the adaptive immune system. Now, all of these, the eosinophils and basophils are involved with various aspects of inflammatory responses. We will have reference briefly to one or two of them when we talk about inflammation, long-term inflammation especially. The last group was the lymphocytes. When we look at the lymphocytes, there are two kinds of lymphocytes. There are T lymphocytes and B lymphocytes. You'll hear about the distinction in a while. We can't tell what they are when we look at them on a blood slide. The monocytes, when they're in circulation in the blood, this is the sort of form that we see. And but in actual fact, the monocytes differentiate into two groups of cells. Both of these are kind of amoeboid cells. And they take up residence in all of our tissues. They do circulate in the body fluids, but they're especially important as surveillance cells. They move through all of our tissues and organs. And the two groups are the macrophages, which are really the most important resident cells in the tissues, which conduct surveillance, and they have powerful communication ability with the rest of the immune system via hormones which they release. The other group which differentiate from the macrophages are the dendritic cells. And I told you this is probably the most important cell of all to think about, and whose function we need to understand. Because the dendritic cells are also surveillance cells, also present in the tissues. But when they encounter something, especially a pathogen, they respond by ingesting the pathogen and then moving to the lymph nodes. They move to the lymphatic vessels and migrate to the lymph nodes. And they convey information from the innate immune system in the tissues to the adaptive immune system in the lymph nodes. And we'll hear exactly how that is done. It's a complex but very interesting process. So here's an actual blood slide stained to show the leukocytes, the white blood cells with their nuclei stained various shades of blue. And first of all, here's a monocyte, easy to remember. A large cell with its very big single nucleus with a crease in the nucleus. This is a lymphocyte here, with its a smaller cell with a nucleus almost filling the cell. This is a neutrophil. The neutrophils are the commonest, and you'll see most of the white blood cells you see in a slide are neutrophils. This, you can see here how this, you recognize it immediately by the strange lobular nucleus here. They're also called polymorphonuclear leukocytes. Simply means nucleus of many different shapes. Self-explanatory. And here is another one here. The other cells that you see here, this is called a basophil. You don't need to remember this, but I'll just tell you that the reason it's called a basophil is because its contents, the cell contents, are basic and stain a deep blue. The cytoplasm stains this deep blue. This is an eosinophil, which is a related cell, but its cytoplasm stains pink. This is its nucleus here, but its cytoplasm stains pink. Both of them filled with granules of substances which will actually cause an inflammation to take place in the tissues. And we'll discuss the process of inflammation in a minute. The rest of these are red blood cells, unstained, they have no nucleus. These here, little fragments here, that's a platelet. And remember, all of these arise from a stem cell in the bone marrow, in the red bone marrow. All our blood cells arise from a common progenitor, a common stem cell. Please, now listen to me, please. When I say that something is important, please put a big note. First of all, you should print out all of these PowerPoints and put a big note says important. You will need to be able to recognize the different kinds of white blood cell from a picture or a diagram. It's not necessarily going to be this diagram, but you're going to need to be able to recognize them and state what their main function is. Okay, so we've mentioned these two parts to the immune system. There are actually two immune systems, if you like, which coordinate with one another, fit together. The first is the innate immune system. All living things have some degree of innate immunity. Even bacteria have innate immunity, are able to respond to the presence of pathogens in an attempt to eliminate them. And the thing that distinguishes the innate system is it's often an immediate response, but it is a response to classes of insult, if you like. So, for example, our own innate immune system recognizes the presence of gram-negative bacteria, and it responds to all gram-negative bacteria in much the same way. It doesn't matter what kind of gram-negative. It responds to gram-positive bacteria all in the same way. It responds to parasites in much the same way, etc., etc. It doesn't distinguish between the individual kinds. And but its real value is that it's a very rapid response. It's a these are very robust responses. We regard our barrier defenses. I told you about the skin, about our mucous membranes, and about all of the secretions that they produce. Mucus containing things like lysozyme, which breaks down gram-positive bacteria. All of these things are barrier defenses, and they are part of the innate immune system. And they are extremely effective. They prevent most pathogens from making their way into that sacred inner space of the body. If a pathogen makes its way across these barriers and enters the tissues or enters the body fluids, then there are a set of responses that are part of the innate immune system. The first thing that happens is that the presence of those pathogens is recognized by phagocytic cells. And the initial most important phagocytic cell is a macrophage. Let me go back here, is the macrophage here. So the macrophage is actually able to ingest and digest pathogens that it recognizes. Also, it can also recognize the debris or non-living things which are foreign as well, and attempt to ingest them and digest them. The second kind, we're not going to discuss much, but they're a hugely interesting group of cells called the natural killer cells. And they are actually, their major task is to recognize the presence of tumor cells and to actually kill them. Because remember that the responsibility, we said right at the beginning, this is one of the major responsibilities of the immune system, is to recognize cells which have been changed in any way, whether it's through infection inside one of our own body cells, or because the cells have changed because they have mutated. The body has these systems to recognize their presence and try to eliminate them. And we are in general very, very successful at removing potentially cancerous tissues, tumorous tissues. Obviously not entirely successful, but by and large, our immune system is extremely successful. And the innate system has these cells, the natural killer cells, whose responsibility it is to do that. The next part of the innate system is the production of very complex proteins whose specific aim is either to limit infection or to kill pathogens and get rid of them, often with the assistance of white blood cells. There are two groups of these proteins that we should remember. The first are the interferons. Interferons are usually released as a result of viral infection, and they are picked up by uninfected cells, which mount a defense to prevent viruses from infecting them. So it's an early warning system, if you like. The interferons have many functions, but that is their basic function. They communicate from damaged cells, if you like, to undamaged cells. The next protein group, it's got a single name, a singular name, which is complement. So complement is actually about 30 different proteins. They are kept inactive in circulation in the blood. They are produced by the liver, but in response to the presence of pathogens and also in response to other immune activity, the complement proteins get together and become active. And they have very strong, for example, they have very strong antibacterial activity. They're actually able to lyse and burst cells. So that is complement. The next thing we'll discuss in some detail in a while. This is the inflammatory response. All of these here can get together to produce a singular kind of response, which is called the inflammatory response. We think of inflammation as being as something damaging, right? We make something bad, okay? We take aspirin as an anti-inflammatory, but in actual fact, inflammation is one of our most important innate immune responses. And we should think very carefully about limiting it too much. So that's the one arm of our our immune system. The next part is the adaptive immune system. And the adaptive immune system is quite different to innate immunity. Because the adaptive immune system is present, by the way, only in jawed vertebrates. And it seems to have arisen at exactly the same time. We don't know why. But the adaptive immune system, unlike innate immunity, the adaptive immune system actually responds to very specific cues. It doesn't just respond to gram-negative bacteria. It becomes able to distinguish between different gram-negative bacteria, for example. It doesn't just respond in a single way to viruses. it is able to distinguish very, very small differences between different viruses. Even very closely related viruses can be distinguished between by the adaptive immune system. And most importantly about the adaptive immune system, it learns to recognize these things, and it remembers what it has seen. So the danger, not the danger, but the disadvantage, let's say, of the adaptive immune system is that it's actually very slow compared to the innate. The innate immune system kicks in immediately, as I'll describe. But the adaptive immune system is much slower. It takes 5 to 10 days to get a full-blown adaptive response, but it forms a memory. The first time it sees something, it's slow. The second time it sees exactly the same thing, it responds again. It responds immediately, almost immediately, much quicker than it did before. So there are two parts to the adaptive immune system. The first is called the humoral response. And this is the production of this very sophisticated, complex set of proteins, which I'm sure you've heard about, especially in relation to the COVID epidemic, which are called the antibodies. And the antibodies defend against infection by interacting with whatever it is, the pathogen, the foreign agent, the foreign substance, whatever it is that is likely, that elicits the adaptive response. The other part of the adaptive immune system is cell mediated. What happens in the adaptive system is that there is a group of cells called the cytotoxic T cell, which can be trained to go out and recognize cells which, for example, have been infected by a virus or by bacteria, or cells, in some cases, which have been transformed into potentially cancerous cells. So they're able to recognize dangerous cells and actually kill them. So these two, this we'll see exactly how it is that the immune system produces this very specific antibody response, and we'll see how these cytotoxic cells are trained to actually go out and do their work. Okay, so this basically I've told you about. This is just a summary of the interferon and complement systems. And we will spend a little bit of time now talking about the innate immune system, especially as a start. The first thing to realize is that the cells of the immune system respond to the presence of pathogens, for example, by recognizing specific molecular patterns that are on the outside of this pathogenic cell. We call those molecular patterns pathogen-associated molecular patterns, or PAMP. And the cells of the immune system respond to PAMP by their own set of receptors called the toll-like receptors. They interact with pathogen-associated molecules, and they set off a response in the innate cell, in the immune cell, rather. So in the innate system, this, of course, this is to classes of pathogen. They can't recognize individual kinds of bacteria, of one group of bacteria. They just treat them all the same. So the immediate response to the presence of PAMP is for certain cells to actually approach the pathogen and attempt to eat it. Now, anything which excites a response from immune cells is called an antigen. Antigen is most commonly associated with an infection, infective cells like bacteria or viruses, but it can also be the new proteins for example, which are produced by tumor cells. So this is the attempt by the response by the immune cells is very often to then attempt to ingest for a very specific reason, not just to kill. In case of neutrophils, it is just to kill. But in other cells, there are other reasons, as we'll see, for why the cells ingest the antigen or the pathogen. I'm going to try and do this. I'm not sure it's going to work. If it doesn't work when I'm doing this recording, then please just have a look at this cell. This is one of the neutrophils. This is a live microscopic film, and let's see if this works. I hope it does. Your screen might be blank. I don't know. I'll describe what's happening. Okay. This is a white blood cell called a neutrophil. These are red blood cells around the outside. These here are bacteria. This neutrophil recognizes PAMP on the surface, and it induces this neutrophil to start chasing the bacteria. Look at it. But look how it eventually is going to catch up to it, and it engulfs it just like an amoeba would. Now it will begin to, it kills it and begins to digest it. So I'm not sure if that worked for you, but I hope it did. If not, go back and have a look at that little movie. There, just think of the complexity of what is happening. When a neutrophil sees bacteria, it starts to chase them. That means it's a single cell. It's got to restructure all of its cellular proteins and everything so that it starts moving. All of its motor proteins have got to get going so that it starts, and it chases the bacteria around and around until it binds them and then forms a fold around the bacteria and actually swallows them and digests them. Okay, so that's the basic activity, but there are some complications to this. Here's a description of how that process takes place. You should remember it from your first semester of biology. This is called phagocytosis. And the cells which can do this are referred to as phagocytic cells. And the most important phagocytic cells we'll see are the macrophages, the neutrophils, dendritic cells that you've already heard about, and B lymphocytes, which you're going to hear about in a while. So remember those four. You'll see them listed again. This is how the fold is formed around the bacteria. This is in response to the presence of PAMP binding to toll-like receptors on the cell membrane. A vacuole is formed around whatever they've eaten. The vacuole fuses with a lysosome, go back to cell structure and look up what a lysosome was and why it exists. It contains digestive enzymes. The two merge together and the digestive enzymes kill off the bacteria and digest them. They are used partially for a nutrient, but the debris is released from the pathogens. This debris may go on to carry information to other cells. There are other cells may pick up this debris and acquire information about the pathogens. But there's another way in which information is conferred. This is very important. It's very important that you listen carefully to what I'm going to tell you, because it explains a tremendous amount about the functioning both of the innate system and the adaptive immune system. It features in both cases. In all cells, cells are able to take parts of the biomolecules which constitute their living processes in their cytoplasm, and they are able to expose them on their cell membranes. They do so, all cells do this, they expose their own innate molecules and on the surface, and they do so with a very complex molecule called MHC, Major Histocompatibility Complex, MHC. And remember this kind of expression of endogenous proteins is done by MHC class 1, and all cells are able to do it. It's a way, in fact, for a cell to label itself as being part of the organism that it belongs to. These are its advertisements to the immune system that this is self. This belongs to the organism. I will just tell you one other thing, because you may have heard of it before. And this is that in humans, this has its own name, which is HLA, human leukocyte antigen. And this is a special name given to human, MHC. Just remember, if you ever see that HLA, just remember, this is what it is. It's MHC. And it's MHC class 1. So this is for all cells. It's their self-advertisement, right? But in a special group of cells, the B cells, which are lymphocytes, dendritic cells that you've already heard about, the ones that wander the tissues and then can make their way to the lymph nodes, and macrophages, when they ingest something and they digest it, they not only spit out debris like this, but they take portions of whatever it is they've digested and they expose it on their surface. And they do it with another molecule called MHC class 2, major histocompatibility class 2. That exposes exogenous proteins or things that have either infected the cell, been ingested by the cell and been digested. That is whatever it is, whether it's bacteria, viruses, whatever, that have been ingested, digested, they can be exposed by MHC class 2. And there is a group of the lymphocytes called the helper T cells that we're going to hear about. They interact only with class 2, but the cytotoxic T cells that we've heard about before, they can interact with class 1 and class 2, as we'll hear. For the moment, mark this as being important. The distinction between MHC class 1 and what they expose, and MHC class 2 and what they expose. So the cells which are able to produce MHC class 2 and expose things that they have ingested and digested, there are these three which are particularly important. They are the macrophages, the dendritic cells, and as we'll hear later, the B cells, the B lymphocytes. Both of these, macrophages and dendritic cells, are regarded as being part of the innate immune system, not of the adaptive, but they definitely interact with the adaptive. And in fact, the interaction of the dendritic cells is critical to the functioning of the adaptive immune system. It carries information from all of the tissues to the adaptive immune system for it to respond. Okay, so in fact, there are these 4 types of really important phagocytic cells, but only those 3 can expose the antigen. Only 3 of them present antigen and are able to produce MHC class 2. The first group of phagocytic cells, leukocytes, are the neutrophils that I've talked about before. These are small cells, incredibly active, very numerous, and they will migrate to areas where they are needed. They will circulate in the blood system, but they can actually leave the blood system and enter the tissues to go where they are needed. They are short-lived, and we'll see their fate in a second. The macrophages, we've said before, found throughout the body. Dendritic cells, found throughout the body. And B lymphocytes. B lymphocytes are made in the bone marrow like everything else, and they mature in the bone marrow, but then they migrate, and they mostly end up in our lymph nodes. They are in other present in other places, but mostly in our lymphoid tissues. That is where they take up residence. And these three, macrophages, dendritic cells, and B lymphocytes, are antigen-presenting cells. They're able to present antigen with MHC class II, and they do so for a very specific reason. Okay, so this is an antigen-presenting cell, and the basic start to the whole process is exactly the same as it would be for any phagocytosis of material from the outside. So we've already seen they form a vacuole containing whatever it is they have ingested. In this case, it's a bacterium that they have ingested. Now, something that's important to realize is that something as complex as a bacterium may contain many, many, many smaller portions which can be exposed. So the digestive process cuts up some major antigen, like a bacterium, into very small antigenic portions. And those tiny antigenic portions are what end up getting exposed on the surface by MHC class II. So you can picture something like a macrophage or a dendritic cell ends up being forested, because they can eat many different things, ends up being forested with these little tiny antigen portions, which are called epitopes. And they are there to communicate to the outside, to the rest of the immune system. Okay, so first digestion, then that gets bound to the MHC class II, and then it gets exposed to the outside. So let's talk now about in the innate immune response, let's talk about how the inflammatory process occurs, which is one of the major responses of the innate system. We take a simple situation. You spike yourself with a thorn. The thorn is by no means sterile. On the outside of the thorn, there are bacteria. You may also put in particles of plant protein and stuff, but we won't think about that for the moment. But on the outside, there may be bacteria. The first thing you've done when this happens is that you've damaged the tissues. And damaged tissues actually release portions of themselves, of their cytoplasm, whatever. And this alone can excite a response from that group of cells that we saw before, called the mast cells. Go back to the diagrams, and you'll see it's on the right-hand side. The mast cells are cells which are present throughout our tissues, all throughout our body. They have a very particular characteristic, and that is when in response, excuse me, in response to damage, in response to the presence of pathogens, they can release signaling compounds. The most important signaling compound that the mast cells release is called histamine. And that I'm sure you've heard of. And because it's histamine that causes responses in allergies, for example. What are most allergies? Just there are inflammatory responses. So just remember that. But if you spike yourself with a thorn, the mast cells respond, first of all, to the tissue damage, which is occurred, but they also respond to the presence of foreign agents such as bacteria. And what they will do is they release histamine, they release some other compounds as well, which are going to cause immediate and very profound changes in the tissue where the damage has occurred. You'll know these immediately. The following symptoms are characteristic of inflammation. The first is swelling. The second is heat, to feel warm. The third is redness. And the fourth is pain. Those four symptoms are the characteristics of inflammation. And many of them are caused by the release of this histamine. Because what histamine does is it causes the blood vessels in the vicinity of the wound to dilate, to increase in diameter so that more blood flows through them. When more blood flows through them, the area will turn red and it will also start to heat up. More blood passing through will heat that area more. The second thing that happens in the inflammatory response is that macrophages, which are present in the tissues as well, respond to the PAMP on the bacteria and begin to release a set of hormones, of immune hormones. They're called the cytokines. And those immune hormones go into circulation. And the first thing that they do is they start to summon neutrophils. At the same time, the walls of the blood vessels become leaky. That is, they allow serum to pass through them into the tissues. That is what causes the swelling of the area. So the leakiness is, first of all, to allow fluid to move in, but second of all, to allow the neutrophils to make their way through the capillary walls and into the tissues. Normally, there are not many neutrophils present in tissue. But if there's an area of infection, the neutrophils begin to accumulate. They are very, very, very active cells. They have a very high metabolism. They are short-lived, but they can eat a lot and kill off a lot of bacteria and debris and everything while they are living. They usually die at the site of the wound, and that is what they accumulate as pus. Pus is mostly dead neutrophils, which have accumulated together with fluid and some fluid and stuff like that. As the neutrophils begin to kill off all the bacteria, macrophages can also eat the pathogen. I'm going to have to take a minute. Okay, so the macrophages are also busy. They are long-lived, though. They don't die off as quickly, and they can move away once the infection finishes. They can actually move away. But remember that they are antigen-presenting cells. We're not going to go into the reasons why macrophages expose antigen via MHC, but just understand that they do, and they do it for a reason. We're just not going to go into it here. Okay, so that explains all of the symptoms of inflammation. Inflammation, first of all, we get the redness because of the dilation. Heat, first of all, because the blood vessels bring more blood to the area and warm it up. The second reason that the heat occurs is because some of the hormones that result from this infection, some of the hormones released by the white blood cells, actually cause the metabolism of the cells in this area to begin to increase. So as the metabolism goes up, so the heat, the warmth of the area increases. And this is advantageous
The immune cells work better at a slightly higher temperature. Things like bacteria, they don't like to be taken above about 37 degrees or so. They don't work as well. Once the neutrophils are killed, everything off, all the healing processes of the skin proceed, and the macrophages end up just cleaning up the whole area. The macrophages end up doing all of the janitorial work of removing it. I do just want to mention one thing, though, that is not present here, shown in this diagram, and that is the presence of these other cells, which are called the dendritic cells. In some ways, they behave much like macrophages. They ingest stuff that they find in this area, but their response, once they have ingested stuff and responded to its presence, is to move to the lymphatic system. And what they are doing, what dendritic cells then do, is they carry information from somewhere in the body where infection has occurred. It does not necessarily have to be bacteria, but they carry information to the lymphatic system, especially to the lymph nodes. And we'll see why in a while. Okay, so important that you know the different steps of the inflammatory response. That's a very localized example of inflammation. So that is the kind of thing you can picture if you whacked yourself with a thorn, whatever. If you do that, you know, if you damage your tissue in any way, you get that immediate response. Okay, so now we need to start thinking about more global kind of responses. And the global immune responses depend very heavily upon the immune system, on the lymphatic system. This is really the communication system of the immune system. The first thing to note is how extensive the lymphatic system is. It goes through all of our tissues, and it's like a parallel circulatory system. It is the lymphatic vessels are structured something like the veins, in that they have valves, so that the fluid is moved through them by muscle contraction, by voluntary muscle contraction largely, but by any muscle contraction will do it. If they surround the vessels, they squeeze the vessels and drive the lymphatic fluid in one direction, and that is towards the heart. The vessels actually empty eventually. You can see them all, they're all flowing up this way through the whole body, down from the head and the neck to these great lymphatic vessels here, which end up emptying into the veins of the heart that enter the heart. They actually enter a vein called the subclavian. And that returns the lymphatic fluids to the general circulatory, to the blood circulatory system through the heart to maintain constant volume. So in addition, the lymphatic vessels actually have portals in them. They have like kind of slits in them, which is how the fluids, which have bathed the cells as interstitial fluid, that is how the fluids enter. So this gives a very easy way for cells like the dendritic cells, for example, to move from the interstitial fluid into the lymphatics and then on through the lymphatic circulatory system. It also gives a way for things like antigens, bacteria, and things like that to move through the lymphatic system. And this becomes important because interspersed along the vessels all the way through, there are filtration stations. That is effectively what they are. These are the lymph nodes. The first thing to note is this is a classic lymph node, sort of a bean-shaped lymph node. There's a kind of lymph nodes you have in your armpits and underneath your chin, or around, but they're scattered throughout the body. But there are many other kinds of lymphoid tissue which are related to the lymph nodes and have very similar functions. There are some entire organs which are part of the lymphatic and
immune system, and these are the spleen there, the thymus gland there, the appendix, and then throughout the gut, there's lymphatic tissue, which we heard about in the circulatory system, called gut-associated lymphoid tissues, GALT. And throughout the mucous membranes, there is a lymphoid tissue called mucus-associated lymphoid tissues, or MALT. And all of these tissues are actually kind of filtering the lymphatic fluid. And they are acting as a center where the cells that are going to produce the adaptive immune response reside. So these here are full of cells associated with the adaptive response. And flowing into them is information from the outside in the form of dendritic cells, for example, bringing information about things that they have ingested, but maybe also bringing in portions of antigen, portions of digested bacteria or viruses. These sorts of things are also filtered here and can be acted on in the lymph node or in the lymphoid tissues. So the one thing that I want to point out as well, we talked about the inflammation as being a response that was relatively local. So it was just this little patch of red and heat and everything else. I want you to picture something, and that is that those responses are taking place in response to hormones that are being released by the immune tissues. But there are other hormones which are produced in response to these situations as well. If stuff goes into circulation in the blood, then, for example, if you get a viral infection or a generalized systemic bacterial infection, then these release of substances becomes universal. Instead of being localized to a little patch where it was, now the responses begin to be generated throughout the body. One of the things that starts to happen is that substances released by especially by the macrophages, but also directly by pathogens, act as pyrogens. That is, they begin to increase the temperature. And one of the things to think about as well is just, if I can just get back here. Just imagine that instead of this dilation of blood vessels being something localized to a little patch of tissue or whatever, instead, the substances which are causing this dilation become generalized throughout the whole body, and all your blood vessels begin to dilate. At the same time, your temperature begins to rise, and instead of there just being localized responses, the responses start to take place throughout your body. If you have a situation, just one aspect of that, if you have a situation in your body where all of your blood vessels suddenly dilate, you can imagine that your blood pressure is just going to plummet. And it may, in fact, plummet to lethal levels. And this is a situation called septic shock. And there's one particular instance where this is particularly dangerous, and that is when bacteria go into circulation in the blood circulatory system, and they cause an inflammatory response throughout the body. All the blood vessels of the body dilate. At the same time, you may have global increases in temperature and all sorts of other responses. At the same time, the blood vessels become leaky. And one of the places where that leakage takes place is into the organs, such as the lungs. And you can imagine all of these, which were localized responses and were quite healthy and not dangerous at all,
Now, if they become global, they can actually become life-threatening. And this is a situation called septic shock. Can occur for various reasons, but very frequently it is due to circulation of a pathogen or of an antigen, which causes a global response of the innate immune system, which can be fatal. Okay, so we're going to leave the innate response behind for the moment, and I'll spend a little bit of time now, in fact, a lot of time, talking about the adaptive immune response, this absolutely incredible, marvelous system, which by and large protects us so incredibly well from the presence of antigen, which could otherwise harm us. And the central to the adaptive response lie the activity of the lymphocytes. Now, the lymphocytes we saw before in diagram and in the slide. Here's another one here in circulation. This is what they look like. A small cell with a very big nucleus like this and not much cytoplasm around it. And there are two groups of lymphocytes. They're called the T cells and the B cells. The T, both of them obviously produced in the red bone marrow, as all our blood cells are. And I'll just show you, there is an organ here called the thymus. It lies behind the breastbone. And the one group of lymphocytes called the T lymphocytes or the T cells are lymphocytes which migrate to the thymus from the bone marrow, and they mature in the thymus. The other group of lymphocytes stay in the bone marrow and they mature there and then move into the circulation and mostly take up residence in the lymphoid tissues, largely the lymph nodes and those associated tissues. So be able to distinguish between the two of them. You can't tell the difference just by looking at them. You have to look at their activity and especially at their surfaces to see what proteins they have in order to distinguish them. They have very different characteristics in their activity. So what does the adaptive immune system actually do? There are four important characteristics. The first thing that I need to explain is that the cells of the adaptive immune system, the lymphocytes, are incredibly specific. They are not just, they are specific to one particular molecular pattern associated with something like a pathogen or whatever. They are responsive to one small portion of the whole thing. They are responsive to an epitope, a small portion, itself an antigen, but part of, usually of a much larger body. That is an important implication. If one lymphocyte can only respond to one epitope, I can tell you we have the capability of responding to about 10 to the 14 epitopes, far more than we will likely ever see in our lifetime or perhaps humanity's lifetime. That is our capability. Now, if that's our capability, it means we're going to have an enormous number of different kinds of lymphocytes. And we indeed do. There are many, many lymphocytes. Their class of activity is much the same, but they're distinguished by proteins which are on the outside, and those are incredibly diverse. They have to be, because one lymphocyte can only respond to one small antigen. So we're going to be exposed to many different antigens. We're going to have to have many different kinds of lymphocytes. The second thing, it's very dangerous for the immune cells to begin to react against our own tissues. They have to be able to distinguish between cells which belong to us and are normal and natural, and cells which do not belong to us, which are foreign, or cells which have changed their nature and may become tumorous or cancerous. But
fundamental to the functioning of the adaptive immune system. The adaptive immune system must not learn to fight our own tissues. If it does, that is a disease-causing situation, and it's called autoimmunity. And as far as possible, the immune system tries to avoid doing that. The third thing that is key to the functioning of the adaptive immune system is, I've said here there's a huge diversity of lymphocytes, but there may be very, very few of them. There may only be one or two that respond to a particular epitope at any one time. But if one of them sees that epitope and is stimulated by its presence, then they begin to frantically clone themselves. And within a very short time, they produce a huge population of clones, each of which is specific just for that epitope. So we go very quickly from just one responsive cell to thousands of responsive cells. The fourth thing, and probably almost the most important thing of all, is that the adaptive immune system is able to form memory. Once it's gone through all of its processes, it actually keeps cells which are immediately responsive. It keeps cells which effectively have a memory of a prior exposure to an antigen. That is the basis of our vaccination systems and everything else, but it's also key to the functioning of our immune system. That is how we become immune to something. We've been exposed to a virus of one sort or another. We may have immunity to that a second or a third or a fourth exposure to that virus and not even notice that we have been subsequently exposed. Okay, so you'll realize this is going to actually become a little bit complicated. I want you, please, from now on, to make sure that you are reviewing materials soon after we do them in class. That means printing out the PowerPoint slides that are on Canvas, sitting with those slides, notes that you make in class and your text, and starting to make careful notes, summaries of what you are learning, not just staring at pages and trying to remember it that way, because you won't. You'll remember when you write the stuff down as if you are discovering it yourself for the first time, okay? We'll tell this whole scenario as a story with a beginning and a middle and an end. We'll define our characters first, and then we'll take them through a plot and see how they interact. The first thing to understand is we've been talking about antigen the whole time. An antigen is any substance which elicits a response from a B or a T cell or other cells of the immune system. In the adaptive immune system, it is anything which elicits a response from a B or a T cell. And the way in which the B and T cells interact with antigen is through receptors, which are present on their cell membranes. The receptors, their function is very similar in B and T cells, but their structure is quite different. So, remembering what I told you, any one lymphocyte can respond only to one antigen, actually one small piece of antigen or portion, which we call the epitope. Here is a B cell. This is a mature B cell, and it is sitting now in lymph node or in lymphoid tissue or something. And it's waiting for exposure to antigen. Here you can see, this is its receptor. We'll see its detailed structure in a minute, but it's a Y shape like this. And at the very end here, it has an area, you could almost think of it being like an active site, where it binds antigen, and it binds very very specifically to only one kind. The whole cell only has that kind of receptor. Okay. The B cells, their receptors have two sites where they can bind. Both sites have to bind exactly the same antigen. The T cell, here's a T cell here, and its receptor works in a similar way in that it has a site here where it binds there. And again, it's extremely specific. One T cell can only bind to one kind of antigen, and very, very specific. So there have to be many different kinds of B cells with many different, each of them with their own receptor. And there are many T cells as well. So here, at any one time, there's only a certain selection of these B and T cells present in the body. There are hundreds of thousands of kinds with it, but they have a certain lifespan and they die out. Our bone marrow generates billions of these in a very short time, and they do it in we won't go into the way in which it is done, but I'll simply tell you that at random, these receptors are generated in many, many, many different forms, so that they can bind many different antigens. Each of the new cells, which is produced, binds one, but billions of cells with different receptors are generated the whole time. So there's a huge diversity. Go back. Remember, that was one of the characteristics that we said of the adaptive immune system. It's incredible diversity. And it's continually evolving diversity, because we do this the whole time. From the bone marrow, we continually produce masses of different kinds of B and T cells. So if we think of an individual antigen, let's say it's a pathogen, like a virus, maybe the COVID virus, for example. If we look on the outside, here's the antigen here, but it has many different kinds of molecules present in many different conformations and everything. Think of that picture of the COVID virus. All of those spikes on the outside, those are all proteins. Each one of those spikes has many different areas where immune cells could interact with them. And if we have something like a virus here, we may have several different possibilities for those receptors to interact there. Here's an important thing. We said that one of the things about the adaptive immune system is that it produces antibody. It produces antibody in response to the presence of antigen. The antibody is nothing more than a soluble form of this B cell receptor, okay? That's all antibody is. A B cell which binds a particular epitope there will start to produce antibody to that epitope. And it does it quite simply by producing molecules very similar to the receptor, but which are soluble. Now there's something very powerful can happen, because we can have many different B cells, for example, which are stimulated by the individual epitope, but they produce antibodies which can coat antigen. So here we have a virus, but the virus, the adaptive immune system has responded by producing two different kinds of antibodies. Why? Because they were produced by two different lymphocytes. This antibody from lymphocyte 1, this antibody from lymphocyte 2. And what they are is no more than a soluble form of this receptor. So this is key to the functioning of the adaptive immune system. We're going to see how we stimulate a B cell with a specific antigen. And in response, that B cell now is going to clone itself into thousands of identical copies, each with identical identical receptors. And each of those is going to then produce masses of soluble form of that receptor, and that is going to form the antibody. In a short time, not immediately like the innate system, but within 5 to 10 days, the adaptive immune system is going to learn about the presence of new antigen. It is going to do so by B cells recognizing the presence of that antigen. And those B cells are going to clone themselves, and they're going to produce masses of antibody. Within 5 to 10 days, we will flood the body with antibody, and we'll find that antibody can actually inactivate the pathogens in various ways. So we'll spend a little bit of time now just talking about the actual receptor itself. We're not going to go into it in enormous detail, but it's important to understand a little bit more about the basics of its functioning. So we'll look first at the receptor. Here I've just drawn in like a cell membrane there. So this is a receptor embedded in the cell membrane. This is a B cell receptor. We know that because it is Y-shaped. It's a complex molecule made up of different polypeptides which are joined together to form the whole functional protein. So this is the diagrammatic version of its structure. Here it is, the Y-shaped receptor, and it has 1, 2, 3, 4 different polypeptides which are united to form the entire molecule, the entire receptor molecule. It's embedded, this is the receptor, so it's embedded into the cell membrane. It has 2 long chains there, they're called the heavy chains there, and it has 2 smaller chains called the light chains there. From here down on the heavy chains and here down on the light chains, we call that the constant region, because for any individual, all of their B cell receptors are going to have this as a constant. But here at the end of them here, these are called the variable regions. And this is what gives each B cell its individual character. Because these variable regions here are generated at random in the bone marrow when these cells differentiate from the stem cell. As they are produced, these variable regions are produced, and all of the receptors on one B cell all have identical variable regions. So they all bind exactly the same antigen. So this whole unit here is identical to that whole unit there. And this is what gives the B cells the incredible diversity, because these variable regions are generated at random. And so this is how we accomplish the diversity. This is how eventually our immune system could react to 10 to the 14 different antigens if you wait long enough. Okay, so here's a process whereby B cells can actually start to produce antibody. And we're just going to picture a simple situation. We have B cells sitting in the lymph node, and antigen, maybe a bacterium or whatever, or part of a bacterium, is swept up to the lymph node by the lymphatic system, dissolved into, floating in the lymph fluid. In the lymph node, it acts just like a filter. You've got the B cell sitting there and starts to concentrate materials and cells which are flowed in through the lymph to enter the lymph node or the lymphoid tissue. A B cell which is able to interact with epitopes or small molecular portions of this whole antigen, they will bind with their
receptor there. If they bind enough, this is key. It doesn't work if you just have one. You actually have to have several bindings like this to stimulate this B cell. If you stimulate the B cell by having enough binding, the B cell will clone itself. And when it clones itself, it changes its form completely. Here it was that small cell with just almost entirely filled by the nucleus with very little cytoplasm. Very quickly, they transform. They form a large cell, which is densely packed with endoplasmic reticulum. Think back. What was the function of endoplasmic reticulum in the cell? It's concerned with protein production. It's concerned with protein maturation. And that is exactly what these cells now are doing. They are now referred to as being plasma cells. There's a B cell which has now expanded, and it's turned itself into an antibody factory. And that is what's happening in this endoplasmic reticulum. It is manufacturing masses of antibody. But the antibody is almost identical to this receptor in that it binds exactly the same epitope, the same little molecular antigen portion. It is soluble, however. It's released into circulation. In fact, the plasma cells leave the lymph nodes and they begin to circulate in the body. If the infection goes on a long time, they will take up residence actually in the bone marrow. But there are now thousands and thousands of identical B cells, plasma cells, all producing antibody to this specific epitope, that one there. They don't produce to this one. You need another B cell to do that. They only produce against this epitope there. The antibody goes into circulation, and wherever it encounters that epitope, whether it's in the tissues, remember the antibody is going to circulate all through the body, all is going to leak out of the capillaries into the interstitial fluids, bathe the cells, move through your organs, go all through your entire body. Wherever it encounters this epitope, it's going to bind to it. And it's going to do it in fact ends up doing all sorts of things. But just as its very simplest, it starts to gum up the pathogen with antibody. And remember as well, you see here, we have this epitope as well. We may have other B cells which react to that epitope. There may be other epitopes on the same pathogen, which other B cells react to. We begin to produce a suite of antibodies which react to all the different epitopes and begin to inactivate the antigen, or the pathogen simply by sticking to it. But that sticking itself may excite all sorts of other reactions. Okay, so this is at its simplest. This is a fundamental response of the B cells. There is a problem though, and that is that a response by the B cells generated like this has some disadvantages. The first disadvantage is that it's not very robust. It's not a very strong antibody response. The second thing is it's not very long lasting. As the amount of circulating antigen begins to fall, the pathogens may be able to rise up again. And one of the reasons for that is that this response produces very poor memory. So it works to a certain extent. And for a small infection, whatever, or something which is very, very highly antigenic, which generates a very powerful immune response, then this might be enough. But it's not enough to give long-lasting protection, especially. But there is a system whereby this initial response is harnessed. And it's harnessed to an interaction with the other leukocytes in the adaptive immune response. and it produces a very robust, full adaptive response with often with good long-term memory. I say often because it depends a bit on the antigen, but in most cases, it produces memory which may be extremely long-lasting. You may be exposed, for example, to antigen in your childhood and still maintain memory against that antigen into your old age. So this immunological memory can be extremely long-lasting. But in order for that full-blown response to take place, the B cells have in some way to interact with the T lymphocytes, and especially with one group of T lymphocytes, which we call the T helper lymphocytes. T helper because they help the B cells mount a full adaptive response. Okay, so we'll end up with this slide and then return to it next class. That will be the Monday after our spring break. So the B and T cell receptors have some similarities to one another in that they both have constant regions and they both have variable regions. The B cell has these two side arms forming this Y-shaped molecule, but the T cell receptor just has one antigen binding site. So this would be this whole end here would be the antigen binding site. In the B cells, this is the antigen binding site there and there. The other major difference between the two, and one that you must remember, is that B cell receptors, as we saw here, are able to bind to free antigen. They can bind to antigen which is just floating in the lymph tissues, the lymph fluids. But T cells are not able to. They have to bind to antigen which is presented by one of those antigen presenting cells. They have to bind to antigen which is presented to them by major histocompatibility complex. And they respond to antigen which is exposed to them by other leukocytes. And T cells especially respond to the presence of antigen presented by those dendritic cells and also by the B cells. And we'll see how this works next time, how this all interacts next time. Please read ahead a bit, even if you don't understand everything. Read ahead, go to these PowerPoint slide sets and read ahead. Read your textbook. Even if you don't understand everything, you will get an idea. You will get a notion about what is to come. And then the class becomes much more elucidatory. It will tell you a lot more if you just even have a vague idea about what is to come. Okay, so then I will see you. I wish you a happy spring break, but I urge you to set time aside during your spring break to go over material and to revise it and to spend time actually studying. Don't just take the whole week off and do nothing, okay? Spend some time actually devoted to revising and to, even if material has been examined already, you want to build a body of working knowledge in your brains, not just have factoids crammed in the night before a test. You need to have working knowledge of this material. And that requires work on your part, okay? So please do that. And I'll see you the Monday after our spring break.